Thanks a lot, and yeah, welcome everybody. It's a pleasure to present here in the ITB meeting. Today I will give a short overview of what we do in my group, and it's mainly physiological based pharmacokinetic modeling, and we apply this in the context of liver function evaluation. So I will jump through many things a bit quick, but I always give references, and if you so the preprints or the references are available, or if you have questions afterwards, just contact me. But sorry, due to the time limit, I have to be a bit fast. So we're mainly interested in liver function, and this is very important because you need the information about the functionality of your liver in the diagnostic, basically to figure out if something is wrong with your liver, and you need it for monitoring disease progression and interventions. So especially if you found something is wrong with the liver and you start some treatment, you're of course interested in, okay, did this work, did this not work, and did I get better, did I get worse? Liver function evaluation is also very important in the context of determining the functional capacity of the liver. And that means basically how much can your liver per volume still do. And this is especially important in the context of hepatectomy, where you cut parts of the liver away and want sufficient remaining liver function, or also in transplantation, where you of course want to have sufficient function after the transplantation, and in the living donor donation, that the donor also has sufficient capacity afterwards. It's astounding. It's very strange, but it's very difficult to actually get values about liver function. There are some gold standards, which are basically liver biopsies, which is just taking a needle and go into the liver and take a sample. And the other are static liver function tests. And these are basically this liver function test parameters you know from your large blood build, it's called in Germany. It's basically this ALT, AST, and these proteins in the plasma or blood, which give you some indication of the liver. The problem is both of these do not actually measure liver function. The biopsies, they give you basically histologies of the tissue and can say if something is wrong. And the static liver function test is basically a plasma marker, which is very poorly correlated with actual liver function. So there's a third option, which is the so-called dynamical liver function test, and this is basically what we are using and modeling in our group. What you can do is that you give a test substance to a person and then evaluate the disappearance of this test substance from the plasma. And this works pretty good because the liver is mainly the metabolic hub of your body and responsible for the detoxification and metabolization of most substances and most drugs. What you then do, you use this rate of disappearance of this test substance or appearance of a product as a proxy for the actual liver function of the patients. There are many different test substances which could be used, and they basically test different pathways in the liver. Examples are indocyanine, green, caffeine, galactose, pravastatin.
Matthias, I think most of us don't see your slides.
No, we don't see your slides advancing at all.
No. Okay. Sorry. Okay. Which slide do you see? Or do you see now
the title slide? We only see the title.
Okay. No worries. Okay. So I have to. Probably I will just reshare my screen.
Okay.
Okay. Yeah. So basically, I just had a slide where said what's going on, and then I showed this disappearance of the curve. Okay. So if you listened, then you're basically like this is the disappearance and the appearance of the curves. Okay.
Okay. Now we can see the rights. Now we can see it changing.
Perfect. And so okay. I think the important thing is like that you have seen this figure here, and this shows the disappearance of an example test substance. So how can we model that? And very nice way to model this is using so-called physiologically based pharmacokinetics models or PBPK models. And what they do, they rebuild the human physiology in silico. And they basically use or combine information from the drug with actual knowledge about the physiology and biology. And what you do, you basically build a compartment model these are mainly ordinary differential equations of the body with the distribution of the substances via blood flow through these different compartments. And depending on your substance or depending on your questions, you put different compartments in here and model the different organs in the detail you need for your question. What you can then do with the such models is mainly simulate the time-dependent concentrations of substances and products in the various organs, mainly in the blood compartments, but also like within the different compartments, like also urine or within the liver and so on. Yeah, here's an example of such curves which you can simulate or can also measure experimentally. And that's basically you give a test substance, for instance, caffeine in a cup of coffee, and then you can simulate how caffeine is appearing over time via absorption, and then how it's basically vanishing from the blood via elimination. And this elimination is mainly the liver, which is converting caffeine to products, mainly paraxanthine, which is then appearing over time. Yeah, and such models are ideal methods for modeling such liver function tests. Yeah, and this is basically what we do. And now I will show a few examples of how we applied this. Like in this case, we were interested in pravastatin, which is one of the statins you take to lower blood cholesterol and LDL-C. And we were mainly interested in what is happening if you have hepatic deficiency or renal problems, and is there some synergistic effect between this hepatorenal impairment. Yeah, the organs which we put in such a model here is like mainly the intestine, which is responsible for the absorption, the liver, which is basically doing the uptake and subsequent biliary excretion, and the kidneys, which are removing pravastatin from the blood. And then we could, basically we build such a model, and then we can change certain variables and just analyze what is happening with the model and compare it to data. And here we basically change the kidney function in different shades of green. Black is normal. The more green it gets, the more renal impairment you have. And in addition, we changed the cirrhosis degree. And what you can see, basically both cirrhosis, which is liver disease, and renal disease have an additional effect and nice dependencies. Yeah, very interestingly, if you look at the total clearance of pravastatin versus the hepatic clearance, we see the renal impairment almost doesn't have an effect, but on the renal clearance, the renal impairment has a very strong effect. Yeah, and then of course we validate. Normally we just validate then model simulations, and our model predictions are in very good agreement with data not used for training. And this is basically one such application. Other things we can do is look at genetic polymorphisms and the effect of gene variants and changes in activity on the pharmacokinetics. We could also do this here with this pravastatin model, and one example is here the ORTP 1B1, which is uptake transporter of pravastatin in the liver. And there's quite a lot of information about changes in pharmacokinetics depending on the genetic variants. And yeah, in good agreement with our model predictions, we see an increase in area under the curve in an inhibition of activity. And if we have increased activity of our genetic variants, then we see a reduction in the area under the curve. So this was also very, very nice. Yeah. Other things we can do is looking more systematically at the effect of such genetic variants. We did this in a model of dextromethorphan, which is, I would say, the poster child of genetic polymorphisms on drug metabolism, because it has such a strong effect. Especially some variants which have basically no activity, and other variants and combination of alleles which have a very high variant, and depending on that, you're either like a poor metabolizer of drugs of CYP2D6, or you have an ultra rapid metabolizer. This is, of course, very crucial in medication of CYP2D6 drugs, because you want to be always with the plasma concentration in the therapeutic range and not too high and not too low. Yeah, so what we here did was they built such a physiological pharmacokinetic model and then coupled it to a detailed model of the different activities from poor metabolizers to ultra metabolizers. And based on these, we could then simulate the distributions of expected pharmacokinetics and pharmacokinetic readouts for populations. Yeah, this works very nice. If we compare the model predictions against simulations, we see that readouts like the plasma concentration of dextromethorphan or of products like dextrorphan, in good agreement, and we can see like this changes in basically in behavior with changing activity over the genotypes. This works also very well for the urinary ratio, and this is one readout which is mainly used for metabolic phenotype. Okay. Yeah, we can take this a step further and then apply this on whole populations. So basically we have a model which allows to this. On the left is the data, on the right is the simulation. So we have a predictive model which allows to, in pretty good agreement, to predict the effect of the various genotypes we have and the various activity scores on the actual pharmacokinetic readout here. And we can then combine things like that with available information on genetic variants in populations, and very nicely also predict whole populations. For instance, here are our predictions for the European and the East Asian populations based on available frequencies of alleles. And this is basically the data which we then later on could correlate. And this shows like within populations, we have large differences in these activities, and the model is able to predict such differences between whole populations. Yeah, other things we're often interested in is virtual populations. Here we did this for indocyanine green. It's a different test substance. And you're mainly interested in, what factors could affect the pharmacokinetics and the readout in models. And which are relevant for the clinics. And what we're often interested in is like anthropometric factors, like how are age, weight, and sex affecting pharmacokinetics, how are physiological factors like blood flow, liver volume, disease, and so on affect pharmacokinetics, and also how surgical interventions change the readouts. Yeah. In this example here, this was a pretty simple model, but we could, in the end, simulate very nicely virtual populations consisting based on actual clinical populations and compare our predictions to the actual clinical readouts. Yeah, it's not important to understand all of that. It's more like it shows like some of the possible applications of such models. And because they're very close to the actual clinical data, it's also very easy to individualize and stratify such models and apply them to predict interesting subsets of target populations. Yeah, I have probably 3-4 minutes more. A key part to enable such modeling and also like the validation of the model is like basically having a good database of pharmacokinetics data. And unfortunately, there's not much data available. And so we went and built a large pharmacokinetic database, which currently consists of around 700 clinical studies and data sets, and around 100,000 data points and so on. And this is mainly what we use to build our models and also to evaluate our models. The problem is often like from your clinical partners, you only get a single data set, but to build the actual models, you need literature data. And this database is basically like the curated literature data, which we use to build the model and sometimes also to validate parts of the model. Yeah. And if you have such a database, you can do very interesting things. On the one hand, you can build models, but on the other hand, you can actually do like meta-analysis and data integration based on pharmacokinetics data. We did this for one example of caffeine, where we created in the end around 140 studies. And then we can look at interesting effects on the pharmacokinetics of drugs. Like for instance, here we looked what is the effect of smoking, what is the effect of all contraceptives of drug metabolization, and so on. And by data integration from around I don't know here 50 studies and so on, we can see clear effects which often are very difficult to see if you only have a single study, and often weren't even studied in the original publications. Other things we can figure out by data integration is things like, okay, that for instance, saliva caffeine concentrations correlate very well with plasma caffeine concentrations. And this is a very nice result because it allows, in the end, basically saliva testing instead of actual blood sampling, which would make tests based on caffeine much easier and much cheaper to apply. Yeah, a big part of our work is reproducibility. On the one hand, we're building such models, but a key feature of these models is that they are open, freely accessible, that all the data is available, and also that our models are all reproducible and openly available. We encode our models in the systems biology markup language, SBML, which is basically a process-based language for ordinary differential equation models. And we enrich that with basically using hierarchical model composition, unit validation, putting annotations on the model, and so on. And this is not only helping in disseminating the models, but also like provides a lot of help in actually building the models or models of high quality. Based on that, we also developed some software. So if people are interested in ordinary differential equation models of processes, please give me a shout out. We have some tools to build SBML models easily from ordinary differential equations. One example is this SBML utils. We also have very nice model reports, which basically mainly we share that with our clinical and experimental partners, or often we do model coupling with other groups and they don't have any clue what we are doing on ordinary differential equations. So this is an intermediate report format which we directly can create from SBML, and we basically communicate via this reports with our partners. There's also visualization of graphs and so on. So I would say, like, if you can get your computational models into SBML, there's a large ecosystem of tools and workflows to make your life modeling easier. Yeah. So one or two last slides. The future, where should this all go here? I would say we want to go towards this digital twins, and this is very popular already in automobile industry and this whole manufacturing sector. It's basically that you build a virtual model which tries to accurately reflect your physical object. And of course, you can do this also for actual patients, and based on measurements, build this virtual populations and virtual people. And a large challenge, or I would say one of the largest challenges is definitely like data availability, data standardization, but also model reproducibility, and and also standard annotations of models and so on to bring data and models together. And in the end, also like evaluation of such digital twins in the actual clinics. But I think this is where it's going. It won't be like I don't know, in the Star Trek and so on, that you have like this full virtual thing in the computer. But I would see this more as it's a question-driven digital twin, which can answer certain questions of relevance for the clinician and the patient. Yeah. Next. Also, other future I see is definitely this digital twins and combining it with artificial intelligence. Yeah, a big problem is with this whole artificial intelligence approaches on electronic health records and patient data, that this is performing very poorly at the moment. And I think a big problem is that we don't have enough, we haven't captured the expert knowledge and the dependencies of variables and features, which physiological based models can do very well. So I think a nice future could be like basically a combination of these computational models, which generate features basically for the AI system, or can enhance features which are already existing in the data sets, and thereby in the end give better predictions. Yeah, very nicely. We got a proposal funded for that, and we will build a demonstrator in the context of liver cancer assessment, basically combining mathematical models and AI, and in the end providing then basically a decision support system for the clinicians. Yeah. Thanks a lot for the attention. Thanks a lot also for all the students which did most of the work, and yeah, of course to all the funders which give money to support that. Thanks a lot.
Thank you, Matthias, for this very nice